My name is Kieran Mitchell. I'm a researcher at the University of Adelaide, and I'm here at the Natural Trap Cave excavation representing the Australian Centre for Ancient DNA. So I'll be doing the DNA work on all the bones that we're pulling out. What we're hoping to achieve by extracting and sequencing the DNA from the bones that we're pulling out of the excavation is to really try and understand what species are present, where those species came from, what the genetic diversity of the species around the Natural Trap Cave was like in the past. And that can sort of help us to understand how the community, the animal community around the cave has changed over the last 30,000 years during a period of pretty rapid climate change. So what I'm about to do now is take a small section out of a bunch of the bones that we've found in the cave. And I'll put those bones into a bag, label it up with the details of where we found it in the cave, what species it was, what element of bone it was. And I'll take all those little bone samples in their bags back to our lab in Adelaide. The first thing I'll do when I get those bone samples back to the lab is to decontaminate them. I'm going to remove all the soil from the outside. I'm also going to remove the very outside of the bone sample itself, which will get rid of any DNA from bacteria or fungus that might have been growing on the bone or in the soil around the bone. And that'll also help to remove any human DNA that might have been transferred onto the fossil during its excavation. The ancient DNA work on the specimens from Natural Trap Cave can really help out with some of the other aspects of the research happening here. What I can do with the DNA is to confirm species identifications that the paleontologists here have made based on the bones themselves. Because sometimes, depending on the bone that you excavate, you may not be able to assign it to a species. You may only be able to assign it to a family level. With the DNA, we can give a very confident identification of the species. One place that this might be helpful is in studying the horse teeth that are being excavated from the cave. One of the other researchers on site is looking at the isotope ratios in the teeth to determine environmental conditions and diet of the horses that were living around Natural Trap Cave. But there were at least 2 horse species in the area. The problem there is that we can't identify which horse species gave us which tooth. So what I can do with the DNA is to identify which species of horse each tooth came from, and then that can help us to look for differences in the diet of the horses. Understanding the diet of extinct species is important in understanding how the climate and the environment have changed, because as climate changes, as other environmental conditions change, the plants growing in the area will change. And looking at what plants particular species are eating can help to explain their distribution or their presence or absence at a given site.